And what you'll see here is that this low level binding is present in a healthy individual compared to this more high level of binding that is present in the Alzheimer's disease patient, indicative of severe inflammation. And what I want to let you all know is that the cells responsible for this inflammation are the innate immune cells of the brain, and those are called microglia. And I'm showing you these microglia here, which are indicated in green and have a smaller cell body and more ramifications in a healthy individual compared to this image on the bottom, in which the individual has Alzheimer's, and we see larger cell bodies with fewer processes or ramifications. And one of the signaling pathways that is relevant to microglia and relevant to understanding my studies is the inflammasome pathway. And the inflammasome pathway is activated in 2 steps. This first step is known as priming, and that is when, in the presence of a misfolded or aggregated protein, in the case of dementia, such as phosphorylated tau or amyloid beta, the pattern recognition receptors on microglia, such as toll-like receptors, are bound and then able to stimulate a signaling cascade that culminates in the nuclear translocation of the transcription factor NF-kappa B. And when this NF-kappa B is in the nucleus, it is responsible for the transcription of proteins related to inflammatory signaling, most notably IL-1 beta and IL-18. And one thing to note is that the second step of inflammasome signaling or activation is very important for the processing of those proteins later. And that occurs when, again, a second signal is sensed, and this can also be phosphorylated tau or aggregated amyloid beta. And this pathogenic protein is phagocytosed by the cell, causing this lysosomal fusion that causes damage to the lysosomal membrane, resulting in the formation of the protein complex that I'm going to refer to as the inflammasome, or eventually ASC-SPEC. And that protein complex is comprised of NLRP3, ASC, and caspase-1. And that complex is able to mature or cleave the previously formed IL-1 beta and IL-18, so that eventually those cytokines are secreted. But more importantly and more recently, we have begun to appreciate this idea that that ASC-SPEC or complex, inflammasome complex itself, can also be secreted. And we know this due to previously published literature by the lab, in which we found that ASC-SPECs compared to healthy controls are actually detectable and elevated in patients with tauopathies. And given this preliminary data and wanting to add to this preliminary data, we actually asked, could ASC-SPECs be measured with AMNIS flow cytometry? And so, to answer that question, we developed a novel methodology in which we processed and spun down cerebral spinal fluid from dementia patients and conducted a staining process in which the sample was blocked with normal goat serum, stained with a primary ASC antibody, washed, and subsequently stained again with a conjugated secondary antibody. In this case, it is Alexaphlor 647, and then we suspended and processed using AMNIS image stream that allows us to visualize the specs and also conduct an analysis in which certain gating parameters can be utilized so that we can actually quantify those specs in patient samples. But before we got to using those patient samples, since they are few and precious, was we validated that idea and that novel methodology a couple of ways. And the first thing we wanted to validate was whether that antibody that we were using, that primary antibody for ASK, was specific. And I did that through the use of Western blot, where I used lysate from an ASK knockout mouse, brain homogenate, as well as patient cerebrospinal fluid samples. And what we found was expected, which is that the ASK knockout sample has no or little ASK, versus the Alzheimer's disease patient cerebrospinal fluid, we see abundant amounts of ASK in all three. And in a healthy control, again, we see no ASK. Again, corroborating what we have shown in previous studies, but also showing that this antibody can maybe be used for AMNIS. And so to test if this antibody could be used for AMNIS, I cultured ASK overexpressing cells, which are specifically a macrophage cell line that overexpresses ASK and also has a cerulean reporter. And when I used those cells as well as the ASK knockout and conducted knockout samples and conducted AMNIS flow cytometry, what I hope you can appreciate is that when we titer the antibody, the purified ASK specs from those macrophages are detectable above the knockout, which is background and indicated by the dashed line. But furthermore, what I hope this graph shows is that when we use the 1:100 dilution, which was also used in the Western blot, what I hope you can appreciate is that the ASK specs are clearly more detectable. And also this idea that even though it's not statistically different, they are almost statistically more detectable at 1:100. And knowing that we could then use this dilution, we also wanted to validate this idea that we could distinguish specs from speed beads, since specs and speed beads are roughly 1 micrometer in size. And what we were able to find through using certain gating parameters and analysis is that the low side scatter population was clearly our ASK specs, while the high side scatter population was our beads. And those populations can be distinguished by those two peaks that I just showed you. And furthermore, this can be corroborated by identifying some example images of what the analysis is picking up. And what I hope you can see is that the ASK spec on the ASK spec channel, we see signal in on the 647 channel versus we see no or low signal on side scatter. While when we want to isolate the beads, we see the opposite, in which those images have little or no 647 signal, while they have obvious side scatter signal. So we were very confident that we were picking out the specs specifically. And these validating experiments made us want to move on with our patient CSF and use that same methodology to analyze human samples. And what we found is that compared to control individuals, people with various forms of Alzheimer's disease or related dementias have an elevated number of ASC specs, and this is something that we are very excited about, since it not only corroborated the previous study, which had substantially fewer samples, but this also allowed us to ask other questions, such as are ASC specs also able to predict other pathologies, including other, are ASC specs able to predict the levels of other pathological proteins, especially given that both ASC and CSF phosphorylated tau 181 are elevated in human samples. So we could ask, what is the actual association between those two proteins if we have that data? And the answer is that ASC specs can actually predict and correlate with cerebrospinal fluid p-tau 181 levels, and also inversely correlate with amyloid beta 42/40 ratio, which is a ratio used to predict amyloid beta load and cognitive decline as a patient. And notably, this 42/40 ratio decreases as cognitive ability decreases as well. So this is very promising. And so I hoped all of this work has convinced you is that ASC specs are clearly elevated in dementia cerebrospinal fluid and clearly correlate with other markers of pathology, such as phosphorylated tau and amyloid beta. And while we are interested in understanding the molecular mechanisms of those associations in the future, I would also like to ask other questions more specific to what I can do with the AMNIS. So, for example, I would like to evaluate ASC spec levels in different patient subgroups, such as males and females, and also use ASC specs to better understand if we can predict the type of Alzheimer's disease or related dementia a patient has, and further through providing a better understanding of markers relevant to vascular dementia, Alzheimer's disease, or mixed dementia. And we would also like to use ASC specs to not only predict those classical pathological markers, but also use those spec levels to maybe predict neurodegeneration by correlating those spec levels with neurofilament light, and also astrocytic damage by correlating these levels with GFAP. And given that the Center for Memory and Aging has a large panel of both neuroinflammatory and vascular factors, we could presumably do this with a substantially larger number of proteins. And with that, I would like to thank my advisors and all the collaborators and members of the Rosenberg and Bhaskar Lab. And I would just again like to thank the NCORE for making this work with AMNIS possible, and Dr. Desai for inviting me to share my work. Thank you all.
Thank you so much, Catherine. That was really, really cool. So on a technical level, one thing that I want to point out is that Catherine's sample is an acellular prep, right? So although classical flow cytometry is very good at phenotyping cells, this is one example where we're actually looking at subcellular particles and being able to leverage the sensitivity of the machine to look at things that are even smaller, as she said, 1 micron, and I'll have an example of something even smaller than that. I would like to invite Dr. Fulong Wang to let us know about his recent publication. He is a postdoc in Dr. Moira Durettich's lab.
Okay, I'm also a user of AMNIS, and I will just add a little bit more about what we can use AMNIS, like what experiments we can do with AMNIS. So everyone knows that AMNIS is like any other cytometry can detect the cell surface receptors. So what I did in this paper is that I detected the CD11B on the neutrophil cell surface to evaluate the neutrophil degranulation. And so this can be so we can clearly see that this AMNIS is a very good measurement of the CD11B on the cell surface, and also you can always like stain multiple cell receptors like L16 in this case. And also I use this to detect the cell surface FPR1 on the cell surface, and we can make all these classic graphs of the immunology aspects. And another thing that we can use AMNIS to do is to detect the secreted extracellular vesicles and particles. That's what nowadays people call the extracellular vesicles, because not only they can not distinguish the vesicles and particles, so they just call them together. So on the left is like how we getting like subsets of the extracellular vesicle particles. So here, in this case, I use CD63 antibodies to scan the EVPs, extracellular vesicles and particles, and we can see that this gating, I don't know whether you can see that as well. So by this gating method, we can see that this category is CD63 positive, and by SSC, it's actually a very low value, so it can be easily distinguished. And here the picture shows like what this CD63 extracellular vesicles looks like. It's very small, but when you zoom in, you can see the dots. And you can easily like give all the measurements of this CD3 positive EVPs by size, by the intensity of the CD63 intensity. And the last thing is that actually we can also use AMNIS to do the co-localization of two proteins inside of the cells using the AMNIS filter. So it's just like what we did in the microscopy for the attached cells. So this is the suspension cells, and here we use the so-called singularity bright detail this time to detect the collaboration between two proteins inside of the cells. And here is the details. Like if the value is like close to 3, it indicates a very high degree of correlations. So we can see in this case in the Y type, there is a very high correlation between two proteins with the exact form. But in the knockout, the correlation is decreased dramatically. So yes, that's it. I just want to learn something again. Thank you.
Thank you. So that's I'm sure that's part of obviously a much larger story and larger paper, but we're very happy to support projects as they contribute to the whole story altogether. So I'll speak on behalf of Dr. Tamara Reutbeck. She's a research assistant professor in the School of Medicine, Department of Neurology. And her focus for her research, and congratulations, Tamara, she is actually retiring this week. So that's why she can't be here. But it's my honor to introduce, the last of her publications where she's really focusing on recovery of stroke. And she's really looking at microRNAs and most recently extracellular vesicles, post-stroke patients. So she came to me a few months ago and said, is this feasible? I'm really new to this field. I myself am very new to it as well, but with collaborations and talks and support from the company and looking at papers, we were able to use, again, like just the way that Catherine did, looking at the beads that help the machine to know the speed of the flow rate and know that they're 1 micron polystyrene beads and be able to gate on objects that are smaller than that, then use side scatter as a way to distinguish between those small events. And then what she's able to show repeatedly is that in stroke patients, there's a higher percentage of particles that show up in her stained channels. Let's see. So the main goal of this whole project is to identify the microRNAs significantly deregulated in stroke samples, which could possibly serve as communicators between the brain and peripheral circulation in human stroke. So all her samples were from peripheral blood. Actually. So that is really just a small snapshot. Again, that study goes much further and looks at many different things, but it was very clear for the distribution and number between peripheral blood, stroke patients and non-stroke patients that she was able to see more of those things. And again, like we saw in Catherine and Fulong's, these are actually extracellular prep, right? So it's very nice to be able to have a different capability in the core with this instrumentation. Guang, do you want me to speak on this? Okay. So I also want to introduce Dr. Guang Huang's work. She's from the College of Pharmacy. So her manuscript that is in the works is looking at arsenite exposure and as it inhibits erythroid development of human hematopoietic progenitor CD34 positive cells from bone marrow, right? Via GATA1 and GATA2 dysregulation and dysfunction. So the first part of it that relates to AMNIS is really looking at the nuclear expression of either GATA1 or GATA2 transcription factor during arsenic exposure, and there's an erythroid expansion and how those differ. So again, doing a series of gating and looking at ultimately at the similarity morphology, she's able to show that cells that have a high similarity score have GATA1 that co-localizes with the DAPI signal, right? Showing that it is in the nucleus. Whereas those cells that fall underneath a low similarity morphology score actually don't co-localize as well. And so this is her way of quantifying whether or not these transcription factors went into the nucleus. So she looked at 3 time points, 5 days, 9 days, and 13 days, looking at both GATA2 up here on the top row, GATA1 on the bottom row, and comparing specifically their nuclear co-localization. So controls are in purple. The treatment is in blue, and what you can see is that there's not a lot of difference when you look at GATA1 nuclear expression, but there is differences in GATA2 expression. So over time, you'll actually see both the controls, but maybe not so much, and then the treatment, the treated sample, there's actually a higher percentage of nuclear GATA2. Maybe not so much difference in GATA1. However, overall, when you look at the nuclear GATA2 expression, there's a decrease over time, and in nuclear GATA1, that trend differs. Am I missing anything? Yeah, good. I don't do you wrong. So this one I think is really lovely. It shows a tremendous amount of work in those figures that. Ever see the figure? This one figure represents 10 years of work. It's not that bad, but it's beautiful where, again, she followed a basic gating strategy and then a more complicated one that was previously published by another group looking at the stages of erythroblasts. So I'm not going to go over that too keenly, but What's beautiful about the AMNIS is not only do you get flow cytometry data with every scatter plot and every histogram, but for every event, you also get imagery, right? So you're looking here at the different stages of erythrocyte development, and it's going from, you can look at bright field from a relatively large cell coming down to a smaller cell, and then you can see the progression and changes of different markers across these stages. And I think it's a really beautiful example of what the AMNIS can do for us, right? Here we're just looking at a broad picture, but I also wanted to say the strength of the AMNIS is you've already seen, we've looked at nuclear localization. We can also look at cell-cell interface. We can look at phagocytosis, cell shape change. So there's a number of ways to use the AMNIS